or whatever the date is today. Welcome to CC Cloaking for whatever the date is today. It is September 29th, I think, 2021. So we have a change of pace today. We're going to go from the astronomy and geotalks that we've been hearing to biology and the early Earth. But before we do that, we are going to do a quick announcement. So I think there's one really important announcement about tomorrow, and Minnie is here to make that announcement.
Hi, everybody. I just wanted to remind everybody that we have our second community conversation of the semester tomorrow, and that will be scheduled at the noon hour, so noon to 1 p.m. And I'll try to provide some updates. Our ADs are going to be here to answer any questions, and of course, we'll have our usual Q&A and interview format that Ariel's been leading, and that should be really interesting as well. So hope to see you there.
Yeah, I'll just put a plug for that. So the Q&A interview thing is going to be with Ashley Landrum, who is a professor at Texas Tech of Science Communication. And she has expertise in particular. She studies people's beliefs in conspiracy theories and how that relates to how people process science information. So it should be kind of fun and interesting. It's obviously relevant to COVID. It's also relevant to all sorts of other things we encounter in our Earth and space science outreach at the world. So it should be kind of fun and interesting and quite relevant also to current events. So with that, I'm really thrilled, excited to introduce my now close colleague and friend and amazing speaker and scientist, Betül Kaçar. And so just a little bit brief sort of standard introduction. So Betül was born and raised in Turkey, where she got her initial education. She came to the US to get her PhD at Emory University in 2010. She then went on to she got interested in astrobiology, searched for life in the universe. She became a NASA astrobiology postdoc at Georgia Tech in Lauren Williams' orbit at Georgia Tech, and then went on to Harvard, where she was a fellow in part in the Harvard Origins program from 2014 to 2017, where she started doing the kind of really innovative work that we're going to hear about today, having to do with resurrecting ancient proteins and figuring out what early life might have been like. She then went to University of Arizona as faculty. So she was an assistant professor at University of Arizona starting in 2018, where she built up a really innovative and vibrant research program around the kind of science you're going to hear about. Many students, many grants. The most recent, or one of the most recent of those grants, certainly the one that's getting the most attention, is she is the principal investigator of a new NASA ICAR award. These are big, those who don't know, these are big multidisciplinary, multi-investigator, multi-institution awards around important topics in astrobiology. And Betül's project, the one that she's leading, that she's a PI of, and full disclosure, I mentioned this in the emails, I'm the deputy PI, so I'm of course a little bit biased. But I was thrilled that Betül asked me to help her run this thing, because what we're trying to figure out is why life uses the chemical elements that it uses. Why these elements? So it's kind of a big question in astrobiology. It's one that, of course, we're going to solve in 5 years. Well, we're going to at least make some serious progress on some parts of it. So it's a very big deal, and it's a particularly big deal to have for an assistant professor, for an untenured assistant professor, to be PI of such an award. And that is a sign of the esteem that the community has for Betül and her research. She recently moved to University of Wisconsin. When we invited her to give this presentation, we thought, oh, great, she's just down the road at U of A, so she'll come in person. But literally like last week, I think, she moved to University of Wisconsin in Madison, and you can see boxes behind her, and she'll probably joke about that. Those are literally boxes from her move like a week ago. So unfortunately, she has to zoom in, but we're thrilled to have her under any circumstances. So Betül is going to talk to us about exploring life's early evolution using ancient enzymes as paleosensors. So with that, Betül Kaçar.
Thank you very much for having me. Oh, okay. Thank you very much for having me, Ariel, and for the great introduction. I wish I was there in person, but another reason for maybe coming back. It's time to talk about metals, because today I will be talking about an enzyme that is very dear to my heart. Many enzymes are, but some are more. And that's not about metal usage, but it is an enzyme that we think allows us to really dig deep into the past and understand, or maybe attempt to understand, the early environmental conditions. So I know that CC particularly, of course, is geared towards space exploration, and you hear many talks from a variety of angles, and biology is definitely one of them. But today really it's just, thanks for giving biology a big chance. And I hope that I don't turn you off and really prove the point that there's a big room for biology in this field, in space sciences, especially when the question that we are asked is at the heart of biology. We are trying to recognize life and forget about different planets, forget about outside of solar system or within our planet. We can not even do this very reliably on our own planet to start with. And that's to me so amazing and really gives the, really conveys the point that biology increasingly, I think, will play a big role in astrobiology, and I'm here for it, and I'm really excited about it. I will have several hot takes, but one of the first one is that I don't think, and I know I'm not the only person here, especially being at CC and scientists that are studying origin of life, that I think not only we need to understand how life on Earth started, but how it evolved over time, or billions of years, and how its early evolution was like, to guide our search for life in the universe. I think this is very important, especially considering that our planet itself been through so much, right? So, and life is a process that's been co-evolving in response to the environment, spanning billions of years on this planet is a part of this journey. And if you think about it, the early systems and early biological systems themselves represent a form of an ancient or alien life form themselves. So in this regard, when we talk about life n equals one problem, or there being only one sample of life, we really need to consider, I think, that the past life may be seen as a way to increase our n equal 1 to n equals to a little bit more than 1, perhaps, especially the more we understand how strange, in a good way, these systems were. I think we will come to an understanding of life as a really big holistic process with all its own quirkiness and weirdness altogether. So, so this is where we started as a group, that we need to understand the generative mechanisms of life to design the best means of life detection. And I know there are many layers between this, but I will try to convey to you how maybe a starting point from molecular sciences, from really biological angle, how this bridge can be formed between life detection and biology. The questions that my group is interested in are understanding the essential attributes of life, and Ariel touched some of this in the beginning. This may be elements, this may be metals, this may be as subtle as a folding behavior of a protein. But how do these seemingly small behaviors and changes or notions impact our understanding of habitability and search for life, in other words? If you think about it, this is a problem of scales. We are trying to find life that exhibits itself if you're trying to find life that will exhibit itself at the planetary scale, that's a big scale, but at the same time, we are looking at molecular behavior. So it is the smallest scale to the largest scale, and connecting these two scales. It's quite fascinating. How do we create molecules that make up for life? We do some work addressing this question, increasingly so we will. That's not what I'm going to talk about today, but particularly, how can we use the information that is available on today's biological systems to create the past life? Can we do this, and why should we do it? And what do we expect to learn out of it from this? So a very quick history of life. Here are the major biological innovations that we know of, and I will be touching on how we know these things and what are the limitations, and how we with today's biology can push it a little further to fill these gaps in our knowledge. So you're looking at life's, the planet's formation 4.5 billion years ago. Life occurred relatively very fast within the first few hundred million years. We have some reason to think that there's biological carbon and sulfur cycling as early as 3.5 billion years ago. Emergence of the oxygenic photosynthesis, all the dotted lines represent our ambiguity in our knowledge, and we don't know exactly when these things happen. We roughly point them to 2.5 billion minus plus. There's definitely some ambiguity there right now, but we have some idea about when these things roughly emerged. And oxygenic photosynthesis obviously is a very big innovation in our past. Before that, there's no oxygen. So when we look for oxygen as a sign of life, we need to consider that for about 2 billion years, there was no oxygen on this planet, yet life seems to be doing fairly well. And then the emergence of eukaryotes, again, roughly 1.6 billion year, and the eukaryotic multicellularity, and then animals, and so on and so forth. So the rest is history, or no pun intended. So this is sort of the big picture, the big innovations when we think about the past. But the question is, how do we reconstruct the unseen molecular history of early life, right? So if you think about it, we are limited, obviously, to what we can reconstruct from the rocks. So, and these are, and I will be touching this in a minute, these are definitely different than finding or coming across to these signatures are different than finding signs of biological life, very different, given the fact that they need to be found and they are not selected by the forces that may be impacting biological selection in any way. And that's also why I think it's quite interesting. How did this, what happened during the first 2 billion years at the biological level that set the tone for life on this planet? This is as deep as understanding the emergence of life's organization, and also finding evidence or understanding how life processed materials and energy to synthesize biomass, and how did this happen in exchange with the environmental structure and changes in the environment. So far, the way we do this as a community, scientifically, we combine, we try to integrate rather 2 different sets of records. At the one level, we have the molecular record. Of course, we are limited to what we see around us today. We build on the extent molecular diversity. We try to interpret the information encoded on today's molecular diversity, either using trees that we refer to as phylogenetics, making sense of the topology, their structure, and try to understand the relationship between today's biological systems, and then try to infer their own ancestry and go to the means of even using them as a way to clock the emergence of certain events in the past. However ambiguous and however limited they might be, this is what we do. At the same time, we have the rock record, of course, we have the fossils, we have some geochemical evidence, we have some reason to think that they can serve as good environmental proxies to understand early environmental conditions, to ultimately connect to what sort of life was present on these locations. So far, we've been really trying as a community to push these 2 records to make sense of each other, especially for the perspective of astrobiology and understanding early life. But I will argue that perhaps maybe we have to also think about, I should say, maybe not rush ourselves to integrate these 2 records and try to untangle them first, perhaps at certain time points, to truly understand what was going on in their own rights, in their own structures, before trying to put the environment on life and vice versa. And I'm hoping that what I said will make more sense throughout this lecture today. But first, let's just take a big step back and look at the hierarchy of life, right? So when we think about life, what do we think about? We may be thinking about trees, or I don't know, plants, or your children, or whatever that comes to your mind when you think about life. Here's an example of the overall structure. I'm using bees as an example for no reason other than the fact that I think bees are really fascinating. So we have the biosphere, the entire planetary ecosystem. And then we have the communities that are formed in these ecosystems. We are drilling down to populations, and organisms make up the populations, and then cell groups form the organism. And when you drill down, we have cell, and then we have molecules. Of course, we can drill this down even further, but our focus as a group is really what's happening at the cell level that is triggered by the molecule, and how did this cellular behavior impacts the biosphere. And you see how many layers we need to sort of drill through to make the connection between a cellular behavior and what happens at the biosphere level. And this is why I think proteins are fascinating, because they bring us this ladder, really, that allow us to climb through these layers and make the connection between the cellular and the cell group and the organismal behavior and the biosphere. I think as a molecular scientist, it is mind-blowing the fact that a single mutation at a molecule can impact not only the behavior of a cell and an organism, but an entire biosphere, to the degree that it may even impact its own atmosphere, to the degree that it may even impact the entire biosignature that this planet may exhibit. I think that is just incredible. So each layer has their own pattern of interaction. We know this. We study molecules, we study ecosystems, we know the rules so far that apply in each layer. And we know that there is some chaos and again, this is not to say that there may be some physicists in the room. This is not a physics verb. What I'm using is more, I would say, chaotic behavior. But at the same time, it's also some machine-like, almost you know how machines are, right? They don't break, they hardly do, and when they break, they're easy to fix. Luckily, life is not like that all the time. So because then it would be very difficult for it to operate. We want some resilience, and machines don't really have that. And each level have some rules that are, again, different than one another, but also similar. And this is, I think, why we can make the connection between a protein and a biosphere, in case you're wondering how are these things are connected. But here are some problems when it comes to understanding early life. And I think majority of us dealing with finding life elsewhere in the universe, or trying to understand how life on this planet originated, or how life was the first, or how ancient life operated, will hear me when I say these out loud, that we need to live with these facts. First of all, when it comes to understanding early life, we rely on very limited morphological data, or any real taxa is really hardly available. So this is a fact when it comes to understanding pests. We have difficulty in tracking the phylogeny and the cladistics overall. So if you're dealing with trees, which we are, to trace the past, this is a problem. The second thing is that there's an organism level upheavals and innovations that happened, but we really don't understand the exact molecular innovations that caused these changes. So these are suborganismal, these could be gene and gene regulatory. We know that these things impact life today. We know the importance of these sequences. You may have heard non-coding sequences, or even the importance of junk DNA. So we know that these sequences are important, but when it comes to early life, we have no clue. And of course, the population level dynamics is a completely closed box right now. And fossils and biosignatures record little of these levels, or at least the way we interpret them, right? We cannot understand a gene regulation change at the biosignature level yet. We are not there right now. The stable isotopes, they're very useful, but they only record a small percentage of functions, of a tiny percentage of a cellular complement. So we sort of accept these limitations when it comes to understanding early life. But here is a fact. Proteins themselves are another record of life, and I think they connect the environmental change and the molecular change really well, and they integrate this really well. They are the powerhouses that gets things done in the cell. And we can, again, use some techniques and methods today to trace their past. So how do we know enzymes extend way back into the past? How do we know when it comes to a protein, we are dealing with something that is billions of years old? And that is, again, biosignatures. That's very, I know CC is very familiar with this concept, that, you know, these could be organic microfossils or sedimentary structures or isotope geochemistry. Even some studies suggested that life could be as old as 4.1 billion years old. Actually, I believe that study came from Wisconsin-Madison here, using zircon as isotope. So we have some idea that these life structures existed back then. So we are really saying is that there was a metabolism, there was a protein or enzyme structures or group of enzymes that carried out these reactions. So when it comes to especially some certain metabolisms like carbon or sulfur, what we are really saying is that something similar to today's biology probably existed back in the day. And because we are using today's biology to interpret these billions of years of rocks, right? So that's sort of what we have to start with, or that's our starting assumption when it comes to interpreting these biosignatures. And you're looking at one of them. This is the carbon. So we have the living microbe, goes through a fossilization process, and then we access, if we're lucky, to these microfossils. If we're really lucky, they're conserved. And if you're really lucky, they belong to Archean. And we have not even a handful of these samples right now. So if you're thinking about it, we're dealing with a few samples to we're writing the story of 2 billion years based on a handful of samples. And in this case, when it comes to carbon, there are multiple isoforms of carbon. Life is lazy. It prefers the lighter isotope. And the way the microbes chew and differentiate between these two isotopes and spit them out gives us a clue of whether what we're dealing with is actually biological or not. We use this technique here, we use it on Mars. This is how we try to interpret whether what we are dealing with is actually whether it belongs or whether it belongs to a living organism or not. So we started with this record. This was really interesting to me, given the fact that it is one of the oldest biosignature that extends all the way back into the past, to the degree that we think carbon fixation is one of the oldest metabolisms that ever emerged on this planet. So it comes to understanding how the behavior of this certain biosignature generation, the carbon isotope, the measurement and the output that I just briefly explained, it's very interesting, because what we're dealing with, and you're looking at this specific carbon isotope differentiation value here over geologic time for different land plants and eukaryotic algae and cyanobacteria going back to the 3.5 billion year record. So what we're looking at is some sort of a uniform carbon isotope value that belongs to organic carbon, and we have the carbonate here for non-living, of course, in our reference. But it may strike to you that it actually looks almost flat, right? And so, and don't let the linear data, the linear structure fool you, because we really have a very handful of, again, data points when it comes to, especially for the Archean. So this to me was interesting for a molecular scientist. So this is the benefit of sitting in geology seminars, that you understand very little. But when a biologist sees this, and that's what I thought, okay, what happened at the molecular level to give this level of isotope variation? And why is this largely consistent, or let me say persistent, over time? Why this hasn't changed? And what are these dips? What are these changes? What happened at the molecular level to cause this? And again, the record gets spottier as you go back, and for an early life scientist, this is also pretty interesting. So then I started asking, okay, how do we actually interpret these geochemical data sets? Again, we compare it to today's modern data. We have the biochemical data, modern values that belong to all kinds of organisms that carry out different carbon chemistries. And we measure their own carbon isotope value, and you may already recognize some of the methanogens that are actually exceedingly different from the range that is present on the left of the figure, right? So, but overall, we try to look at today's microbes and obtain a value, and then try to map it on the geochemical data set that we have in hand in order to make sense of the past record. So my question was, what caused this? What is thought to cause this at the molecular level? And at that point, it turned out to be a protein. And tada, so not surprising, because proteins are awesome. So here is the protein Rubisco. And this was a very interesting story for me, because I always thought Rubisco, especially when I was in college, one of the most intimidating enzymes ever. And I could not escape from Rubisco. And I thought, this is maybe in my destiny that I have to study this enzyme, because I keep running away from it and it keeps finding me. So Rubisco is the enzyme that is thought to be one of the prime mediators of the carbon discrimination today. That's what we study, that's what we see. And it generates a difference in the carbon isotope record and dominates the ecosystem today. It's the highest expressed enzyme. I put it to the scale of the planet. It is the highest expressed protein on the planet. It's a target to many synthetic biologists, many bioengineers that is trying to improve its function solely for the fact that it plays such an important role for carbon fixation and photosynthesis. At the same time, it's a very old enzyme, right? So when, again, we extend the carbon fixation and the emergence of carbon fixation based on what we extract from the rock record to billions of years into the past. So we assume that the same value exists today. Okay, it must be a similar metabolism. And so I know it sounds extremely simplified, but that is really the thought process, and that makes a lot of sense. Obviously, we have to anchor it to something that we understand and know. So Rubisco is an ancient enzyme. We think something like Rubisco existed in the past. And what's really interesting about this enzyme is that it catalyzes the rate determining step in Calvin cycle. And you see what I'm saying? In college, learning about Calvin cycle and this enzyme was Not so easy for me, but here we go. So we have and Rubisco can uptake both carbon dioxide and oxygen, and that's a problem, of course, because we are dealing with an enzyme that's at the heart of carbon fixation. And in the case of breathing oxygen instead of carbon dioxide, instead of Calvin cycle, the enzyme catalyzes the downstream photorespiration reaction, and that leads to a major biomass loss. There's been a lot of studies that calculated how much biomass is lost or how much they can be gained also by subtle tweaks on this enzyme. And it has not been very successful. We are so far not very successful in improving the specificity of this enzyme towards CO2 so it stops accidentally reacting with the oxygen instead. But this is also another interesting fact about this enzyme. If this is as old as we think it is, which is about 3.5 billion years old, when there was no oxygen on this planet, when did this specificity issue arise? And how did the ancestral Rubisco behave? Was it more specific towards CO2 or not? So we started with this, and again, knowing that enzymes interface with the environment and their host organism, we started to think about whether we can answer some of the questions about Rubisco using our own tool set. These questions were, first of all, how old is Rubisco? And we don't know the answer to this. What was the function of ancestral Rubisco? We don't know. And how consistent is the Rubisco-mediated carbon isotope fractionation value over evolutionary time? We don't know. Even though what I showed you very briefly is thought to be caused by an enzyme like Rubisco or Rubisco itself over billions of years, we have not tested these things and really asked these hard questions when it comes to understanding our own assumption of the past. Even though, again, we map the modern phenotype onto the past to interpret these ancient isotope signals. So this was very interesting for us, right? So here we are, we have a system, we have an enzyme that is very interesting and is very crucial when it comes to our interpretation of the past, yet we don't know the very basic questions when it comes to understanding itself. So with that, we set a study to reconstruct the ancestral Rubisco. We said, okay, let's bring this enzyme back, let's resurrect it, let's bring the oldest version to the lab and then study its behavior. And obviously, the first thing we're going to do is let's test whether the ancient Rubisco will actually give us the same carbon isotope fractionation value or not. And as I said, the record gets spottier as we go back way back in the past. That means that if we actually create the system, perhaps we can use these ancient modern hybrid systems to fill these gaps. So we are really trying to use biology to resurrect what is recorded in the rock and maybe what is not and what could have been. So this is our way of attempt to synthesize life as we don't know it in the lab by using synthetic systems. And our approach is very interdisciplinary. NASA likes to call this a smoothie, not a fruit salad. It is very true. We use evolution as a start. We use the information that is recorded on modern systems and generate evolutionary trees. And sometimes resurrect the ancient DNA after our inferences directly. Sometimes we only carry out computational studies, but the majority of the time we do experimental work. We then engineer the ancient DNA that we synthesize in the lab inside the modern organism genomes. So we sort of genetically modify modern bacteria with ancient DNA sequences to see how this bacteria will behave in response to this ancient DNA. It is like a Jurassic Park, if that's what you're thinking, in a reverse way. And then we connect, the ultimate goal is to connect what this ancient modern hybrid organism, what it does to what is recorded in the rocks, to finally interpret their behavior at the global scale. So the key for us is to find those the connector points and proteins that impact the plant trigger a planetary level behavior. So of course, this took me a really long time, years really. When I was a NASA postdoc, this is what I studied, and I dedicated years in the lab to create these ancient modern hybrid systems. You can imagine all kinds of problems arose when it comes to pushing these microbes to take and synthetic alien DNA. but it works because I'm here. So here we have the artificial proteins to reconstruct ancestral biogeochemistry. That's the direction we are going for right now. We have the protein sequences to start with, we align them. I'm not going to drill down the details. I'm happy to talk about it if you wish later, but we create phylogenies. Again, we mathematically infer the ancestral node, then we synthesize it in the lab, and then eventually engineer it inside modern organisms to finally use them for a biogeochemical interpretation. And this is a new system that we established. So what I'm going to talk about briefly is that how we use the first step for creating the ancestral version of the Rubisco enzyme. And there you go. So we published this, so I'm just going to skim through it. But basically, we create phylogeny trees using all the forms that modern Rubisco forms that are available today, and then create a phylogeny tree. We use modern sequences. I'm happy to talk about the details. There are a lot of tweaks. It takes us a few months really to come up with the best topology. We do a lot of different mathematical tests, and then run different alignments and run different models. And we've lately been powering these things with a lot of machine learning algorithms to make sure we come up with the most reliable and realistic sequence, because not all of the sequences are very, we are not all confident at 100%, of course, when it comes to reconstructing the past, but we try to come up with the best, most likely ancestral gene sequence that we can, and then reconstruct the ancestors. We always make our ancestors publicly available, so anyone can access these things and download, and that will make us really happy. So the idea is to start with the youngest proteins and then trace our way back into the past. So you're looking at a 3 billion year old history here, just by following the branch lengths on the tree. So the first question was, does the Rubisco sequence change in response to the rise of oxygen on our planet? Because we could access billions of years of variants of Rubisco, we focus on certain spots on the protein. And then because there are a lot of, again, studies, biochemical studies, biophysical studies, when it comes to understanding the behavior of this protein, we rely on the literature a lot. So this is really great. These are very well studied enzyme systems. And then see if we see changes in any of these particular regions that correspond to the carbon dioxide oxygen reaction site. And I'll go through this rather fast, but the answer is yes, that there are changes on the enzyme sequence itself, that for the enzyme versions that correspond to the great oxidation event. And the reason why we think that is about as old as the GOE is because these are usually the ancestral cyanobacteria on our tree. So we don't necessarily date the tree, date the emergence of the metabolisms ourselves, but rely on the formerly interpreted clocks or generated clocks. But what was interesting is that you're looking at 5 different protein structures here. They may all look the same to you, which is very interesting, because what we see is that this enzyme looks pretty much the same over billions of years of time, which is not so surprising when it comes to biology. Biology does really amazing, surprising things, but at the same time, it manages to do that while doing really boring things at the molecular level somehow. It's really interesting. So at the structural level, the enzyme looks the same, but at the sequence level, it's not. So even though these changes are very subtle, they may be subtle. Seems like, you know, they aren't, and I'll come to why I think that way in a minute. But we saw that some of the sites that are important for carbon dioxide were actually different, which already told us that no, this enzyme was not the same in the past. And that prior to the rise of oxygen, the enzyme may behave differently. So the idea of mapping this protein on the ancestral states and assuming that its function was the same or similar throughout billions of years may not be completely true. So this is also quite interesting because we are seeing an information of the environment signed on the enzyme, that the enzyme is telling us, that the ancient versions are telling us that I was responding to a change in the environment. Here is my site, here is the amino acid site that I have that is reacting with the oxygen, and it was different in the past. I think that's quite something that these are whispering us about adaptation that they had to an environmental change billions of years ago. So, and these are also, you know, we refer to them as signatures of primitiveness sites that were very primitive, but again, then changed in response to the environment. If you are interested in a more philosophical take on this, we published a paper discussing what this may mean. But moving forward, we wanted to understand what's the function. And this is the first question I asked. So we want to resurrect these genes. We don't want to just make them in the computer. We want to bring them back to life. So let's go with this. And this was what I did in Arizona. And we resurrected this gene by doing two things. We created them, we cloned them inside the plasmids, and then we developed a system to directly engineer the cyanobacterial genome by using these ancestral variants and doing genetic engineering. So the experimental setup was overall pretty much like this. It's going to be going clockwise, just stay with me. But here is the organism. What you need to know is that we basically found a site on the cyanobacteria that is not impacted from the circadian rhythm. So these are always on genetic sites. And then we scissored out the entire operon of Rubisco, the modern one, swapped the modern with the ancient DNA, made a new fragment, and then engineered it back to the neutral site. So this was really we didn't use CRISPR, but it is a similar basically procedure using enzymes that are not as fancy, but they still do the same thing. Basically, they are molecular scissor enzymes. You can think of them that way. After that, we obtained these enzymes. Of course, this took a lot of screening, a lot of design. It's really, we have to put engineering hat here. And then we moved on to characterization of these ancient modern hybrid organisms, finally measuring their behavior and carbon isotope discrimination. So I want to present these results. These are in review right now. But we used one of the youngest ancestor. We usually do this. We don't start with the oldest, most different variant, but start with the relatively younger. And here is the variant that we picked as the ancestor of modern 1B, if there are Rubisco experts in the room. But and you see how we constrain it. It's very broad. That's us being very cautious. We don't know exactly how old this variant is. But you may realize that we put it to Precambrian. Basically, we're dealing with a variant that is definitely at least 500 million year old. But it's difficult to, and I don't want to directly age this thing when we don't know exactly when it lived. But the cool thing was that this thing is alive, right? So we could take the synthetic ancient DNA, put it inside a modern organism, and generate a bug that is living in front of us. So that was quite a day. So you were looking at the first round of data that we generated. We moved on to growing these things in different conditions and under different atmospheric conditions, under air or a little bit higher CO2, just to mimic early environments a little bit, especially the pre-GOE. And again, we're dealing with the ancient one, that's the Enc1b, ancient 1b, and we have our system here. We created multiple strains as control. So we have a strain that is only, we call these things wild type in biology. I don't know why. And but that's what we do. And then this is a synthetic system one, where we have basically we modified and put the wild type, the native gene inside the neutral engineering domain that we found in the genome with the help of Susan Golden's lab at UCSD, and also the strain that only harbors ancient Rubisco. So on the bottom, you're seeing our carbon isotope discrimination results. Again, these are not published yet. This is on the bioarchive, but we have the air, the carbon isotope discrimination value under air and under higher CO2. So you may realize that the ancestral one, at least for sure for the air, discriminates carbon a little heavier compared to wild type and these two wild type variants. However, all these values matched with what is interpreted from the rocks. So even though we are getting a little higher discrimination value, we are able to replicate what is also inferred from the rocks. So at least when it comes to 1B, we were able to repeat what is coming from actual fossils. And we also carried out growth and some photosynthetic evolution experiments using this system. Overall, integrating this ancient DNA slowed down the growth and organisms' growth capacity, as well as the Rubisco activity, meaning that the ancient enzyme is not as active as the modern one. This is usually a problem that we encounter with dealing with ancestral systems. that they are not optimal. And of course, you may think that on the entire machinery, the rest of the machinery is modern, you're only changing one single component. That is true. And that's exactly why we're picking the enzymes that we think are the prime dominators of these metabolisms. In the case of carbon fixation, and for this particular bug, Rubisco is, at least it's thought to be, and it is catalyzing the rate determining step. So it's catalyzing the slowest step in its own essential reaction. So to mitigate this, we run multiple control experiments and we create multiple strains. And I can't drill down into this, but we've done a lot of genetic testing to make sure that what we observe isn't an artifact of a slow growing bug, but is really truly can be attributed to the ancient Rubisco itself. And if it is not the case, we try to discern at what point we may attribute this to the ancient component versus making a system that is simply malfunctioning due to perturbation. So just to wrap up, we are observing a distinct carbon isotope fractionation value to corroborate this the uniformity and assumption, at least when it comes to this system. So this is very interesting that we created a system for the first time using synthetic biology and apply it to biogeochemistry for the interpretation of the rock record. So on its own right, I think this is very, it is for us, it's a very big step for moving forward to use these systems. And we're happy that NASA also seems to agree that we are going to be doing a lot of this work in the near future. But of course, it's important to understand that there are many fundamental attributes of this extant and ancestral metabolisms. And they play a role in the biosignature production overall. And we have, again, these are yet to be characterized. And I reviewed some of these things early on, all the subcellular dynamics and all the non-regulatory region impacts and all the gene expression impacts that geneticists are very familiar with in today's systems. There's no reason to think that they didn't play a role in the past, yet we don't understand them and how they, again, impact our understanding of interpreting biosignatures. And really distangling them will be very important, I think, for our own interpretation of our own life's history on this planet. So it is it's sort of a, again, this is not a talk that will give you a lot of answers, but maybe we will leave it more questions. that how do we know the past biology was not more different than was the same? I don't think our starting assumption should avoid this question. We should always think that it may be more complicated than what we think it is. And when it comes to understanding what these bugs ate or how they lived, how old they are, it's all very complicated. So, but at the same time, you know, we have to accept that the cellular environment there's a homeostatic control in its own terms, by its own environment. And I was just rambling these thoughts, but cells, we should consider them in their own isolated environment, subject to their own regulatory feedback and limitations before we push the change of the environment on interpreting what the cells are doing and how they recorded themselves on the rock, right? So this is, I think, important for our true understanding of the biological and the geologic records, that we need to, I think, untangle them and decouple them at key points and really resist the urge of combining molecular record and the geologic record and writing these just so stories that just because there was oxygen in the environment, this happened, or just because there was certain metal in the environment, life took that metal. That's not how I think it works in general. I think we need to be careful with making these assumptions on life itself, but it's definitely more complicated than an environmental availability or a direct environmental reservoir impact and impose. So I'll say, let's just decouple these things and evaluate them separately. And that is what we did originally with Rubisco, and that's what we're doing moving forward. We are now inserting even older ancestors and try to understand how far we can take this our own fossil records that we created using live organisms, however paradoxical that may sound. And that to really give protein level innovations a chance and develop these experimental systems to understand early innovations. And here you're looking at not Rubisco, but nitrogenase, another enzyme that we're working on that this time prefers different metals. So it connects us to the biosphere level change that I was pointing to by using different parameters. And just to wrap up, this is also what Ariel mentioned early on. We are really growing. It's quite exciting. This whole molecular paleobiology theme to really reconstructing the past to understand it and study it. However, you know, it's not, however imperfect our system might be, this is a start. And as long as, you know, we have the health and energy and funds, we will continue to push this. There's a lot of different systems we can apply this to, so it's quite exciting. One of them is nitrogen fixation. We launched Muse last week. It's the metal utilization and selection across eons, our own ICAR. You can visit our website. And this is the entire theme is to study biological innovations across eons, Earth's past and present and future. In this case, focusing on metal use. And I presented what we're doing with carbon, but also expanding this to nitrogen accusation and see how we can build on modern systems to push biology a little further and see what life is capable of. And finally, modern organisms and their genomes are the products of contingent evolutionary trajectories. This is my final big message. And it's not always clear to what extent the modern biology and its characteristics represent their ancestral predecessors. And again, this is not something we should be afraid of. This is something that should motivate us to push this further. Thank you very much for having me.
Thank you very much. So people who have questions online, and there's more people online than in the room here, please type your questions in the Q&A. That's the easiest way for us to keep track of them. We'll also take questions from the audience here. If there are questions, raise your hand. Otherwise, I will start asking questions, but it's better if students do.
Hi, so I have a question. I think you might have mentioned this in your talk, but I just want to know how the enzyme Rubisco has been naturally preserved over the geological time record. Like, that was like pretty curious to me as to how it hasn't even been altered structurally.
Yeah, so thank you for asking this question, as it allows me to clarify a very important point. We cannot extract the DNA sequences from such old rocks. I think the oldest we can go is really maybe a few 10,000 years from permafrost. And that is really not sufficient for us because we're interested in deep time. So when it comes to deep time, we cannot extract DNA. So that's one thing, we don't do that. Two is we are using mathematical and evolutionary models to interpret, to infer and then synthesize and then interpret ancient DNA. We assume, the third question that you asked, we assume that Rubisco was the prime player in shaping the isotope record because we use what we create, right? So the data we generate using modern organisms is what we use as a proxy to interpret ancient rocks. And we have an enzyme today that plays a predominant role. It's important to note that there are several carbon fixation pathways. There are many different enzymes that also play an important role. However, this seems to be the one that is more frequently visited, Rubisco. That's why we picked that one, and that's why we use that one as one of the thought to be important enzymes when it comes to shaping the past also. And structurally, this is something we generally observe with essential enzymes. They tend to evolve slowly, and you know, that makes sense, I think, given that they play an important role. And structurally, we observe this for other ancestral essential enzymes too, that they tend to retain their structure over time. It's quite fascinating. Thank you.
Let me ask a question from the online Q&A. Are there non-enzymatic reactions that fix carbon that predate Rubisco, and are there therefore theoretical proto-Rubiscos that are simpler in structure?
Yeah, that would be, that's very interesting. That probably, if they're non-enzymatic, I'm not sure if they are abiotic, right? So that would be interesting. But when it comes to an enzyme that is capable of doing what Rubisco is doing, I won't be surprised if there are proto-Rubiscos that may be even simpler in many ways, that were capable of doing the job of this huge enzyme. So I won't be surprised for sure. That's a good idea.
So if I can ask a follow up to that. So how far back can you push this, right? So you pushed it back to a certain extent. How much further back could you push the evolutionary?
That's a very good question. So so I wanted to I refer to this as early life because we can expand our reach to the degree of what we can put on the tree, right? So our root, our deepest branch, that's usually what determines how far we are. And this is definitely not origin of life. At this point, we are assuming that we are post-Luca. And so the question before you, that is, I think, what they're thinking about too, that this is definitely, they're asking pre-Luca or pre-tractable system. And that's the exact problem about Archaean too. It gets, again, it's not tractable when it comes to past, right? And we lose that tractability pre-Luca too. So we actually rely on synthetic systems to see what we can create. But yeah, in our systems, usually we are limited to whatever root we have on the tree.
So is there another question in the room?
Hi, so I was wondering what would be some of the more difficult challenges to recreating ancient Rubisco enzymes?
Technical challenges? Or
Yes.
What were they? Well, we're still tangling them. Number one thing is, I think sometimes these ancient genes, they don't express really well. So the cells make them, but not enough. And usually essential proteins, essential enzymes are really highly expressed in the cell. And Rubisco is a very highly expressed protein. So it's a success in its own right to diminish its expression. So for we didn't do it for the results I presented, but that's what we're doing now. We add artificial promoters to promote the expression of the genes artificially to just help us give the cells a little push to make a lot of them. But usually, yeah, that's usually the problem, that they are so old and diverged that the modern cells don't recognize them, that they don't work well with the modern components. That's usually the prime challenge.
Thank you.
So can I just ask a follow-up from that? I mean, that right away, right, tells you that there's other things that have changed in the cell, right? And so when you're trying to characterize physiologically how these ancient enzymes work, since other things in the cell have changed, it's not really a fair game, right? You don't know how it worked in the ancient organisms. So how do you? Yeah. I mean, I know you can't deal with that right now, but how do you imagine dealing with that?
So
in the future?
You gotta pick your battles, right? So we may go to creating an entirely ancient metabolism. We have not done that yet, or a pathway that is all synthetic and old. We are very reduced, and we are hoping to grow this more now. But our initial steps were single. But I must say that again, usually, and this is related to the previous question, that it's usually the amount of the enzyme that seems to be the problem. And that's what we go for. In fact, some of the changes that we observe in, say, 2 billion-year-old Rubiscos, the changes are located on the enzyme structure that actually impact the expression. So biology, I think, uses expression as a sort of brake, or gas pedal that either increase the amount of protein you make or decrease it. And changes at the gene coding region are not very prevalent, I think, when it comes to essential systems, because you can imagine they're problematic. That I think cells want to protect their essential players. Either changing the amount or the decreasing or increasing is a good strategy to deal with the environmental upheaval.
Right. So this is kind of a first step towards eventually being able to resurrect entire metabolisms or organisms, right?
Yeah, and this is, we developed these systems. We did this a lot with translation machinery that I didn't talk about today. And we've done a lot of work on really discerning to address the question before you, that what will be the problems? What is the problem because it's an ancient gene, and what are the problems because we are messing with the system? And they are not always the same.
All right. There's another question in the room.
Yes. So in regards to Rubisco and the GOE, I know results are still kind of preliminary with the evolution of Rubisco, but does it seem so far that the influence in that event happening was more environment driving genetic change or a mutation than heavily affecting the environment or affecting the environment?
Okay, that's a very deep question. I don't have the experimental result to tell you that. However, our GeoBio paper where we looked at the Rubisco structure sequence, it seemed to be, maybe this is a stretch, but there is a tiny branch right before that we think that coincides with the GOE. And that also coincides with the sort of whiff of oxygen hypothesis that that subtle changes in the atmosphere before the rise of oxygen, or before the major event. And that is also when we see the changes that correspond to the regulation region of Rubisco. So we think maybe it is to realize your first suggestion that it could be that environment draw the enzymes response. But again, we haven't tested that in the lab, so I shouldn't say either way.
So Professor Everett Schock has a question.
So this is the...
Oh, you're answering his question? Should
Yeah.
I ask her? Let me ask her first. So how do amino acid abundances change with time in reconstructed Rubisco?
In amino acid abundance, you mean the change of amino acid abundance over geologic time, or the?
Yeah, I think it means the abundances of the amino acids of the various amino acids in the enzyme.
So we haven't correlated that with the amino acid abundance change overall. We are doing that actually for nitrogenates today as a part of the ICAR. But the amino acid change is about 40% when we compare the modern Rubisco to the oldest Rubisco. So we see about 40% difference in amino acid sequence itself. But I haven't characterized whether whether there's a trend in the amino acid characteristics per se. Here I'm showing you the alignment between the modern Rubisco and the ancestral 1B. That's the youngest ancestor that we resurrected first. And that's about, I think, 20 or so, 24 amino acid difference overall.
All right, so I imagine if Everett was in the room, there would be some back and forth here around his question. In the interest of time, I think we should probably go about wrapping it up. Is there a final question in the room?
I see two. Okay, sorry. Go ahead.
There are a couple of questions. There are a couple of questions in the Q&A that I think reflect some slight misconception of what you're doing. I don't know if you want to address those here or if we want to address them in writing later. It's kind of up to you. Do you see them?
I can address them unless there are questions in the room.
No, go for it.
So the question is, are there specific sites that I collect in order to get proteins of interest to do my research? So we don't bias our selection. We rely on today's information. And when we create these trees, we are we try to be very careful not to bias the tree towards a certain protein, towards a certain families, meaning we don't over choose from cyanobacteria, otherwise our ancestors would resemble that. So we try to represent all taxa equally, which is not always easy because not everything is sequenced equally.
But you're not sampling physical locations, right? You're grabbing.
I'm not sampling physical locations. We are relying on what others sampled, and we use their sequences from the database.
So I mean, sometimes simple questions reveal like really interesting things. So how do you know that what's been sampled, right, that you're sampling from is actually representative of, how do you know there aren't really interesting life forms out there in strange places that? Exactly.
So the more diversity there is, the better for us. We keep our trees very diverse. We have to. And if we don't, there's a reason for it and we report it accordingly. And but what really determines is the alignment, our initial alignment that determines the quality of our reconstruction. And this is why we spend months, because you can make a tree in 2 days, but do you make it right? That's the question. And also all trees are wrong, right? So that's important to accept. And but some are useful.
Right. So you know that these reconstructions are not quite right, but hopefully they're right in their essence.
I don't think we can. I think I need to be out of my mind to claim that we absolutely are dealing with the true ancestor that existed billions of years ago. I mean, that would be insane to say, right? So we rely on the mathematical inference and our statistical confidence, and we are as confident as our numbers are. So I cannot claim that we deal with the true ancestor. We don't have a way to test that time moves forward as far as we know. So we cannot reverse the clock in real life. I have to travel in time and actually get these samples from Archean and then bring them back and sequence them, which would be cool. But no, so.
Yeah, that's for somebody else to figure out.
But they are all in the papers. That how well we know them. We of course report all of those.
All right. So I think we do need to wrap it up. We'll leave it to somebody else to invent time travel to do this absolutely correctly. But meanwhile, this is the best way we're going to do a molecular time travel through statistical models. So thank you very much, Betül. That was great. That was a very fun talk. Thanks to all of you here and all of you online. And we will see you next week.